Well, perfect. We're going to go ahead and get started. Welcome, everybody, to Phil's last official Field Crops Virtual Breakfast. Thank you all for being here this morning. We're going to hear from Dr. Sarah Hughson, our arthropod, insect and arthropod diagnostician of the MSU Plant and Pest Diagnostics Lab. So, Sarah, take it away.
All right, thank you guys for having me today. So again, my name is Sarah Hughson, and I am the arthropod diagnostician in the MSU Plant and Pest Diagnostics Lab. So today I'm going to talk to you guys a little bit about what we do in the diagnostics lab, a little bit about insect diagnostics and that process, and then I'll share a few insects that I've received into the diagnostics lab. So at Plant and Pest Diagnostics, our mission is to assist our clients in identifying and solving their plant and pest problems. And so we can do that in a number of different ways. We can do that by identifying arthropods, so insects, mites, and ticks. We can do disease identification, nematode quantification for soil samples in the field. We can do plant ID for weeds, and we can look at some different abiotic issues that folks see in the field, like environmental issues and herbicide injury. So what type of samples do we work with in the lab? Well, we work with basically every type of plant sample that is grown in Michigan. And I'll also accept some of the household insect samples as well. Now, from this pie chart, you can see that many of the samples we receive are field crop samples. So 49% of our samples are field crops. And the vast majority of those are going to be nematodes for nematode quantification in the field. We also get some plant disease type questions, but actually very few of the field crops questions we receive are insect questions. And so that's one of the reasons that I wanted to speak with you guys today, to just let you know that we can look at your field crops insects as well. So that is a service that's available to you guys. And actually my background is in field crops insects. So I do really like to see those samples when they come in. Now, when you're submitting a sample, the way you can do that is just by collecting some of that affected plant material, putting that in a bag and sending that to us. If you include some soil with that, that's great. We can look at that soil for pH and other parameters. And if you guys are able to email some pictures of the field, that's even better. That can help us determine kind of the distribution of the plants in the field that are affected and how many plants are affected in that field. Now, if you don't have the plant material, but you have collected the insect, you can also send just the insect. So you can do that by putting them in a vial of alcohol, just tossing them in a jar or other hard-sided container, or sending in a sticky trap that has your insect on it. Now, with your sticky traps, one of the limitations with that is if the characteristic I need for identification is on the side that is stuck to the sticky trap, then I don't necessarily see that structure. But I can usually still give you treatment recommendations and get close to an ID. So what do you get from us? So when you submit a sample, you're going to receive a diagnostics report. That's going to include the identification of the insect. That's going to include some relevant biology of the insect and some control methods for that insect. I do my best to answer all the questions that the client has about the insect and to try to anticipate some questions that maybe the client doesn't know to ask about that animal. And if you submit a regulated pest, then we can also assist you in reporting that pest. Okay, so let's talk about the diagnostics portion. So when a sample comes into the lab, the first thing that I'm going to do is put that under the microscope, whether that's a plant or an insect. And I'm going to look at the characteristics of that, and I'm going to look at the injury that I see on the plant matter. So what am I looking for in an insect sample? I'm going to use this nice little cutworm as an example here. So what I'm looking for with insects is a unique combination of characteristics that are unique to that individual species or a particular group of insects. So that could be things like the antennae on the insect. So here I've got the little cutworm, and you can see one of its antennae here on the side of the head. I might be looking at things like the mouth part of the insect. Does it have chewing mouth parts? Does it have sucking mouth parts? I might be looking at the legs. So here I have an example of the prolegs of this caterpillar, and you can even see the little crochets that it uses to hang onto the plant. I might be looking at things like wing venation on wasps or on flies, and I might be looking at really tiny structures on these animals. So for very small insects or very young insects, I might be looking at things like the placement of hairs on the body or the placement of very tiny spots and markings. And so this is an image of the side of this caterpillar. And I actually used the placement of the speckles on this caterpillar and the placement of hairs to decide which species that was. Now, when I'm looking at plant material, what I'm looking for is the type of feeding injury on that plant material. So I might be looking at chewing injury that can help me narrow down which groups of insect I'm looking at. So even if I don't have the insect, if I see skeletonization injury, I know that that's probably a caterpillar, a beetle, or a sawfly. I can also look at discoloration of the leaves. That's usually an insect with a sucking type mouth part. I can be looking for cupping and curling of the leaves, leaf mining, galls, or I can look for materials that the insects have left behind, like webbing, cocoons, or shed skins. So now I'm going to show you guys a couple of field crop samples that have come into the lab. One of these is from last year, and then two of them just came in the past week. But I think these are interesting samples that you guys will like to see. So this first one was from last year. This is an example of black cutworm on corn. And for the diagnostics on this, what I'm looking for is that this is an early season pest. These caterpillars are active at night. So a lot of times the client doesn't see the caterpillars in the field. They just see the damage to the plant. For that damage with the very young caterpillars, we can see some small holes in the leaves on the young corn. And for the older caterpillars, what they're going to do is actually wrap their body around that stalk and they're going to chew that plant clean off. So when you look at the plants, they're going to look like they've been cut off with a knife and they're going to be cut off at the base of the plant where it meets the ground. And so this is an image that the client submitted along with their physical sample. And you can see that that plant is cut off right at the ground level. Now, another thing that's great about these client images from the field is that we can get more information about the field. Now, with black cutworm, we know that the females really like to lay their eggs in crop residue or in weeds. And what we see here in this image is that we do have crop residue right around this cut plant. Okay, so we actually have multiple things that we can see here to identify black cutworm, with the main thing being that cut plant. Now, when we're controlling for black cutworm, we typically don't use preventative measures for black cutworm. This is a sporadic pest, so a lot of times, your efforts would be wasted in doing a preventative treatment or preventative methods for this insect. But if you're really worried about black cutworm, things that you can do are just cultural management, like removal of that crop residue or weed management early in the season. Now, with black cutworm, when we do treat for them, we usually do a rescue treatment. And we can apply that rescue treatment when our threshold is reached, 2 to 5% cut plants in the field. So that's going to be stand loss in that field. All right, I'm going to show you guys our next insect, or actually this one is a mite. So this one is spider mite on corn. This is a really interesting one that came in last week. And this one was actually on sweet corn. It was not on field corn, but it's kind of a similar situation. So in this case, the client was concerned about the gray discoloration on these leaves, and they also saw some spider mites on the leaves. So when I look at these leaves, the first thing that I notice is that there's quite a bit of yellow stippling on the leaves. So this speckling here, and that's what we usually see when we have spider mites on plants. But we also have this kind of fuzzy gray material. So was that a fungus, something like that? I asked Jan Byrne, our plant pathologist, and she looked at that and she said it did look like a fungus, but not a type of fungus that would harm the plant. So we looked at this under the microscope and we did see spider mites. So I have an example of the spider mite on the picture at the top of the screen. And then the picture on the lower portion of the screen, you can see there's a lot going on the surface of this leaf. So all of these smaller yellow spheres that you see on the surface of this leaf are spider mite eggs. So what we've actually got going on this leaf is like a sheet of spider mite eggs across the leaf. So at some point they had a ton of spider mites on this plant. What we also see is that we have this fungal growth on top of those eggs. So we have a whole nother layer of material here. And, Jan had said this isn't a type of fungus that harms the plant. So I think what's happening here is that this is one of our beneficial fungus types that grows on insect eggs and spider mite eggs. So with humid conditions and with frequent rains, that really promotes fungal growth, those beneficial fungi. And so I think what's happening here is, yeah, we have a ton of spider mite eggs, but then we have a lot of this fungus growing on the plant. And that's something that might help destroy those eggs. So this particular client may not need to treat at all because they have this beneficial fungus growing. So we'll talk a little bit more about spider mite management. So again, in this case, the client has this beneficial fungus growing on these mite eggs, so they probably don't need to treat. But some things to take into consideration when you do have spider mites. I've got 3 points here for that. The first one is to remember that this one was sweet corn, so our management might be slightly different. With sweet corn, this showed up around the time that people might be harvesting sweet corn. So I think if this was very close to harvest, they probably don't need to treat. It might be too late, and if they were to treat, it might be a wasted effort because they're about to harvest that. If these plants were a little bit earlier in their development, if they were in that time period where the plant is shifting from vegetative growth over to reproductive growth and over to that point where they're filling those kernels, that might actually be a good time to treat because we do want to make sure that the plant has all the energy it needs to fill those kernels. A couple other considerations with spider mites, these guys often impact plants along the edge of the field, so the injury can look a little bit worse than it is. So you want to make sure you scout into the field to make sure you still have that issue. With a lot of our animals that damage plants along the edge of the fields, it usually doesn't have an economic impact. So it might be one of those things that looks bad, but isn't really going to impact you. Another consideration is how much stippling you have on the leaves. And these leaves did have quite a bit of stippling. But for some of the plants, they can handle quite a bit of stippling without having a huge impact. So again, that's another one of those things that you want to make sure the stippling, there's heavy stippling into that field before you treat. And then our third insect here is going to be Japanese beetle on soybeans. I received an email from a client asking what to do about these Japanese beetles. So when we're identifying Japanese beetles, the adults are going to be pretty conspicuous. They're going to be on the surface of the leaf. They're not really going to be hiding from you. You can identify these guys because they have the brown metallic wing covers. They have kind of a green metallic head area, and they're going to skeletonize these leaves. They tend to feed more on the upper canopy of the soybean plant, and they also tend to feed along the edges of the fields. So this is another insect that maybe doesn't have an economic impact on the field all the time. This is one where you're going to want to scout and make sure that you have a lot of defoliation into the field before you treat. Now we have two thresholds for Japanese beetles, and these thresholds are actually good for all of the defoliators in soybeans. So if you have high percentage of foliar damage, this could be Japanese beetle, or it could be from bean leaf beetle, then you can still treat for those insects. Now, if you have 30% defoliation before bloom, you can treat. And if you have 20% defoliation after bloom, so this would be the period of pod fill for those beans, then you can treat. Now, we have some challenges with our Japanese beetles. And the challenge is basically that Japanese beetles are always challenging. Regardless of your area of agriculture, you may have problems with Japanese beetles even after you've treated for Japanese beetles. These guys are extremely mobile, and even if you kill all the Japanese beetles in your field, they can move from other locations into your field to feed on your plants. So one of the questions I frequently get from clients is, I've treated for Japanese beetles, I've done all the things I'm supposed to do, and I still have Japanese beetles. What can I do? And the answer to that is that, you're doing all the right things for Japanese beetles. You just have to keep doing them because these guys can be difficult to deal with. So it's continued monitoring. And that's the last insect I have for you guys. So I'm going to say thank you for your attention. If you have questions about plant pest diagnostics or would like to submit a sample, please feel free to reach out to us. And so with that, I think that's the end of my time.
All right, so this is a question kind of combined for Jeff and Marty, but will these cool temperatures that are coming up trigger tar spot?
Hey, Nicole. So yes, tar spot is heavily driven by temperature. So in all the modeling work we've done, temperature is a key driver. So these cooler temperatures are very favorable. Any tar spot that is there, during those warmer weeks that we did have may not have been very active. It would be sitting there and it wouldn't be killed by those warmer temperatures, but just waiting for temperatures to cool off. So yes.
Thank you. Jeff, how long can we expect the smoke to stay in Michigan?
The current episode that we have is fairly short. We've been through most of it right now. The northern part of the state actually has had smoke since the early part of the beginning of the week when it first the jet stream started to change. But for most of us, it should be wrapping up in the next 24 to 36 hours, at least the noticeable haze. And it's enough. The concentrations are enough for the Eagle with the state here to issue an advisory for help for particulate matter. So it is a little bit higher concentration, but I think it also is short duration. Given the medium range forecast guidance, though, that we looked at, one would have to expect that there won't be after this episode. We probably will go a while before, but at least a couple weeks before seeing smoke again, because we won't have that northwesterly component that we do now.
Great, thanks, Jeff. Moving on to our insect experts. How many generations of leafhopper can we have in Michigan, particularly for dry beans and alfalfa?
Yeah, so I answered the question in the chat. It takes about 10 days for eggs to hatch and then a couple weeks for the nymphs to develop through their stages. And then you get adults and they, mate and lay some eggs. So you're looking at, roughly 25 to 30 days. So say like a month if you want to round it up. And leafhoppers tend to come in May. So you'd have a generation in June, a generation in July, a generation in August, maybe a fourth generation if it's warm. So we're probably Phil said he saw some damage in alfalfa recently that probably hadn't been cut yet. We're probably into the second sucking generation, and then we're working on a third. Now these cooler temperatures and a little wetter, if that can kind of keep going, there's a beneficial fungus out there that kills potato leafhopper. It's free and it's in the environment. So that, depending on the weather later into August, if you're in a droughty area though, potato leafhoppers can be pretty bad because their feeding interacts with that droughtiness.
Great, thank you. And then Chris, you had posted some updated regional thresholds. Do you want to just cover those?
Yeah, so we had funding in the last cycle of the North Central soybean grant. So this is the checkoff dollars coming from Michigan and all the other places. They go into the North Central soybean research. There's a big acronym for it, but they had funded an we've had a regional project for many years since the beginning of soybean aphid. And in the last few years, what we worked on was our defoliation thresholds, because people asked, shouldn't they be updated? And that sort of thing. So a bunch of states got together, we toiled away at that. And the new defoliation thresholds, we allow a lot of defoliation in the vegetative stages and then the early flowering. Once you get to R3 and R5 and you're filling pods, then it goes down to only 10%, and then back up to 15 for R6. And then after that, after R7, you would not spray for defoliation. So, and that would be for all defoliators, Japanese beetle, beanleaf beetle, caterpillars, whatever is out there. And the edges of fields, as Sarah showed, those edges of fields are always bad for Japanese beetle. They're mating right now on the edges of fields, and they're lazy. So they'll go in from a turf or a ditchy area and sit on that edge and then eat and mate at the same time. But as you walk into those fields, that defoliation really trends off really fast. So those are updated thresholds, and I'm updating the management guide right now for the fall. So that those updated thresholds will be in there, along with a nice explainer of how to rate defoliation that isn't so hard as just your eyeballs. So stay tuned for that for the winter.
And also, I should clarify too that the question that I received about Japanese beetle was from a researcher. And I've actually never seen a field situation with a grower where they needed to treat for defoliation for Japanese beetle specifically. I think that researcher was just trying to eliminate those Japanese beetles as a variable to their work.
Yeah, that makes a lot of sense. I know that the soybean breeding program can see differences in their feeding across their lines. So sometimes they have to treat for leafhoppers or defoliators to remove that, as Sarah said, as a point of their yield.
Yeah, and I should say, so because I don't receive that many field crop samples yet, I would like to receive more. But because I don't receive that many at this time, the ones that I do receive tend to be kind of oddball samples, and questions.
Thanks. And then I have a question for you as well. Um, when we're looking at insect identification, and let's say that you find something in a field that you wouldn't generally expect to see there, what are kind of the steps that you take to then identify that?
Yeah, so in some cases, folks can send me pictures of the insects that they find. But if your insect is something that's very young, like a small caterpillar, or if it's something that's very
Hey,
small, like the mites, it's actually better if they send them into the lab. But if it's something unusual, I might actually go through the process of identifying it with a dichotomous key. So I've actually trained to site ID a lot of different types of insects. So for most of your major pests, I can look at it and I know what species it is. But if it is something unusual, then I'm going to have to get out the big textbooks and I'm going to have to go line by line and look at those characteristics to figure out which critter we're looking at.
Great, thanks. That's maybe something I'd say generally most of us don't want to be doing out in the field. So thanks for doing that
Yeah,
for us.
yeah, and
A question for, sorry, Sarah, what did you say?
I said it can be difficult to do that in the field because oftentimes you need a microscope.
Gotcha. A question for Marty. Will application of fungicides help corn and soybeans deal with drought stress?
That's a bit of a tricky question to answer. There is a little bit of data to show some benefit to that, but I would not be applying fungicides for drought stress personally. So if we think about return on investment, we really need to be spraying for disease. That's where we see the greatest potential for that return on investment. So in the links there, I posted a return on investment calculators. So we have one for white mold on soybeans. You can look at different products and how they perform, and one for corn fungicides as well. So that pulls in all the university data, not just Michigan, but Wisconsin, other states around the area that you can look at and play with. I'd really urge you to do that. And if you're spraying a fungicide, again, please leave check strips. If you're just spending all this money and not doing your homework to figure out how it's working out for you, probably not the best thing to be doing, especially in these tight markets.
Great, thanks, Marty. Phil, you've got your hand up. Is there something you want to say?
Yeah, I wanted to ask Jeff about cutting alfalfa or cutting hay, and what about the relative humidities, temperature combinations over the next couple weeks? A lot of guys need 3 to 4 days minimum to get hay dry, and so it's always a concern. Are we going to see a heavy dews with these lower temperatures too, that are going to
yeah,
delay our ability to dry hay?
good question. And it's a little bit of two-edged sword here because the daytime conditions are going to be ideal. Low humidity, strong sun, really good drying rates. But at night, we're very likely going to be seeing those nighttime, where the temperature drops off. There will be an extended and heavy dew likely to form with low temperatures. And so obviously at nighttime, there's gonna be re-wetting of some of the forage on the ground, and not ideal. And we're going to be going through at least four, maybe five of these cycles of that. So good during the day, but at nighttime there will be a little bit of a setback because of the cooler weather conditions. Now, as we move into that medium range time frame that we looked at with the warmer and wetter, we would be seeing more with elevated temperatures, the evaporation and the drying would be a little bit enhanced given no rainfall. That would be again looking into the second week of August. But for the upcoming week, you might be lucky enough to get things done and not have a drop of any precip on it. But the nighttime dew cycle is going to be a little bit of a hindrance because of the type of conditions we're gonna deal with.
All right, thanks.
And then some questions here on sending in samples. can you send ethanol through the mail? You cannot because it's flammable, but it sounds like there's something else you could use. So Sarah, you want to speak to that?
Yeah, you're welcome to use just white vinegar. It doesn't preserve the sample as well, but yeah, USPS is happier if you use that.
Thank you. And then another question, how accurate is the Picture Insect app?
Yeah, so a lot of the insect apps out there are not very good. But I'm trying to test out the Picture Insect one recently, and I think that this is actually the best one. So I've had it bring back correct answers, pretty regularly. I've had it get some that where it's kind of close, and I've had a couple where it was kind of way off. But I think for the most part, that particular app does pretty well.
Great, thanks. And then it looks like from a couple comments in the chat, yeah, the more pictures you take, probably the easier it will be for it to identify.
Yeah, probably.
Okay, well, with that, thank you, Sarah, for your presentation. Thank you, Jeff. And of course, you know how to reach us all, and Sarah shared her information. I've dropped the link to the Plant and Pest Diagnostic Lab in the chat. And yes, best luck to you in your retirement, Phil. And we thank you all for coming to the virtual breakfast today.